This session is Paola Bermudez. So I'm going to read her short bio sketch, which is just as written here. Paola Bermudez is a final year PhD candidate in the tissue engineering and mechanobiology lab at the University of Bern, Switzerland. Her research focuses on key metabolic events involved in intervertebral disc degeneration and identification of potential anabolic mediators for IVD regeneration. Paola is claiming that distinguishing these phenotypes could elucidate fundamental metabolic mechanisms in IVD degeneration and generate novel targets for therapeutics. Paola has been graduated from the University of Navarra, Spain, with bachelor's degree in chemistry in 2017 and biochemistry in 2019. And then she did her master's in biomedical research with specialty in the regenerative medicine and advanced therapeutics. So Paola is going to give us a talk on the title, Modulating Intervertebral Disc Catabolism in vitro and ex vivo systems, IL-4 and IL-10 metabolic role in intervertebral disc degeneration. So Paola, this is your time.
Okay, thank you for the kind introduction and welcome to my presentation. So as you can see in my slide, I am going to talk again about the intervertebral disc, as Andrea already did very nicely. But in my case, I will focus more in the extracellular matrix metabolism, and I will also present different culture systems to study this matter. So going back a bit what Andrea already mentioned, we are part of this For All grant, and we are tackling the low back pain, since it's an extremely common condition experienced by people of all ages, and it's considered a leading cause for disability worldwide. Also, it's a high-cost condition in our society. It could be even comparable to cancer disease or cardiovascular disease, and it has multiple contributors. But rather than talk about maybe a bit boring epidemiological data, I decided to bring you today some more cases about low back pain that I experienced in the last years. So one of it is the case of Rachel Boderman, which is this one. It's not the one of the middle. And she was my master's student together with Chloe in the spring in Bern, and she did the elective module of her master's in biomedicine in our lab. And one of the reasons why she decided to do the master thesis there was because she was a patient that suffered low back pain for a lot of time, like in her past. So basically, one day she woke up with such an acute low back pain at the age of 18 years old, like really young. And she went to the doctors, and they were really not paying that much attention. She was a young, healthy woman. It was like, well, it will be something, muscular maybe or so, you will be fine. But she was really dealing with this acute low back pain through over a year, and she ended up having an MRI scan because she really was not able to really have a normal life anymore. And once they got the results, they saw that she had a huge inflammation in the lumbar part of her spine. So then they thought even that she could have maybe an autoimmune disease or something related to the immune system rather than something related with the spine. And she spent also several months without diagnosis. She was tested every week for such a random, strange autoimmune disease, and always negative, until an orthopedic surgeon took her case, and he decided to do a discectomy, which is basically remove the disc between the two vertebrae. And she got that surgery at the age of 19. And once she got that, she was healthy again and pain-free. This is like the first case that I am bringing. And the second one, it's a more personal and a special case for me because it's the case of my cat. It sounds a bit funny and like stupid in certain way, but animals can also suffer low back pain and also have intervertebral degeneration. In fact, there are even animal models that they are used for studying this kind of this disease, since they naturally develop this disease. Like, for example, the beagles, this breed of dogs. In the case of my cat, we observed that she had such a strange behavior. She was not running anymore. She was not jumping. And she was arching a lot the back, the spine. So we took her to the vet, and after an X-ray, she was diagnosed with a spinal fusion, which is already when the discs are that degraded that the vertebrae they start fusing between together. So she totally lost the mobility of the spine in that region, and like it was diagnosed in that way. But now, once I already showed these cases, I'm going to go a bit closer and talk about what the intervertebral disc is. So as Andrea already mentioned, it's located between two adjacent vertebrae in the spine, and it's responsible for the flexibility of the spine, the multi-axial motion, and low transmission. If we look to more in detail which are the different regions and tissues that we can find in the disc, we have in the center the nucleus pulposus region, which is proteoglycan-rich region, meaning that these proteoglycans are negatively charged and they keep all the water inside, making it really look like soft. And everything is peripherically surrounded by the other region, the annulus the annulus fibrosus region, which is more fibro-like region. And both of these regions are encapsulated between two adjacent cartilage end plates that are the responsible of the nutrient, oxygen, and waste transport. Because the disc itself, it doesn't have vasculature, meaning that it's an immunoprivileged organ, as Andrea already explained. And that also makes it a really harsh environment, meaning that the oxygen and the nutrients, the glucose, always reach through diffusion. So in the center, it's a really hypoxic environment. The glucose is also really low, but the cells are like used to this kind of environment, and this is how they work in a physiological way. If we look a bit closer to the nucleus pulposus tissue or region, which is one I have been studying during my PhD, we can see that there are different players in it. We have nucleus pulposus cells, and then we have they are surrounded by extracellular matrix. For example, collagen type 2, collagen type 4, also peptidoglycans. For example, we also check a lot the aggregan content, which also tell us how healthy is the extracellular matrix. And also we check, for example, other growth factors like the TGF-beta. And all of these, we consider them anabolic factors. But of course, we also need to have a balance, and we need to have some catabolic ones that are, they are in charge of renewing all the old extracellular matrix and making room for a new one, which are, for example, the metalloproteinases or the ADAMs. They're in the picture. And also we have source cytokines that are more catabolic, like interleukin, tumor necrosis factor, interferon. And those are considered catabolic markers or factors. Of course, when a disc starts degenerating, all these features that I have been mentioning, they totally change. The first thing that we start observing also in the clinics is like the disc height starts decreasing. That means that the capacity of the nucleus pulposus of keeping water it's degrading, and all the boundary between these two tissues start getting like lost. You cannot really see where was the MP, what was the AF. That also brings the loss of mechanical properties that they had at the beginning and they stopped having. And of course, the metabolism in those tissues it gets really unbalanced. And that leads, of course, to nerve and blood vessel ingrowth inside the disc, starting it can reach even the nucleus pulposus, but here in the picture I have it in the annulus fibrosus. And that basically leads to pain, degeneration, and more disability. So again, if we zoom in again in the nucleus pulposus, but in this case in the degenerative one, we will see the cells start clustering. They even go under apoptosis. The cells start getting like lost in the tissue. And then we can see all these catabolic factors, they really start increasing, all the interleukins. Also, the ones that are in charge to push the nerves and blood vessels inside, like the neural growth factor, avascular and endothelial growth factor start increasing, and of course all the matrix the proteins in charge in degrading the matrix start also getting unregulated, making the catabolic factors really important in the environment. And if we check, in this case, the anabolic ones, we start seeing that how they start degrading. We can see here the collagen 2 and the proteoglycans start degrading because of this unbalanced metabolism, and that makes the anabolic factors be less important in the tissue. But of course, we are in the human digital twin summer school, so we need a bit of bioinformatics and models, not only biological data. And a thing that Jeroen mentioned about it was that we need to find a connection between models and real tissue. And I had the opportunity to work during my PhD really close with Sofia, that I think that you might know her. She's giving the hands-on number 5 together with Francesco. And she basically developed a knowledge-based network model of the nucleus pulposus tissue, where she mapped different catabolic and anabolic factors, and then she checked which could be the most relevant ones, which could be the potential anabolic mediators that we could think about them as a regenerative therapy. And after she developed all this method, she ended up with some predictions, like the IL-10 and IL-4 being potentially anabolic markers. And that was taken to my project, and then we established some aims like investigating the metabolic events in human cultures and also trying to understand the role of IL-4 and IL-10 in the human IBD. So this is my study design so far. I'm going to go part for part because I think it's a bit dense to see it like this. But basically, as a first concept, just to mention that I have like two main parts. One, a proof of concept part, where I wanted to see if IL-4, IL-10, and their receptors were present in the human tissue, in nucleus pulposus tissue. And the second part was to start stimulating the cells with all these both cytokines in different cultures models, in vitro, ex vivo, and also using different cell types considered healthy because they come from trauma patients, meaning that someone is paralyzing in the Alps, they're back in Switzerland, has an accident, it's taken to the hospital, and they need to remove a disc, but it's not because he or she had in the past a degeneration, it was because she or he had an accident. And then we have the degenerative ones that they are coming from patients that they have been suffering this degeneration, and it has confirmed that they have this disease. But first, let's check the proof of concept part, where I basically ran the immunohistochemical analysis that Andrea really, really nicely explained the methods. So you already know this. I don't need to explain it. And basically, after optimizing the protocol and kind of selecting what was the best antigen retrieval method and primary antibody concentration, we conclude that they were present, IL-4, IL-10, in the human nucleus pulposus tissue, as well as the receptors. So physiologically, we have them in our spine. Then I also had the opportunity to work with the QPath tutorial that Andrea and Maria Paola prepared really nicely. They made it easy for biologists, which is like a plus. Thank you so much for that. And I was able also to go a bit deeper in the analysis. In this case, I checked the immunopositivity rate of IL-10 receptor, and I also checked how it was distributed in different age, genders, IBD structures, this level, and spinal pain. I did not see any difference between groups, and we could really say that it's quite kind of evenly expressed with a mean of expression of 42%. So it's quite expressed in the disc. Then, once we confirm that these cytokines are present and the receptors are present, the next part was to try to see what role they have in the disc. And for that, as I mentioned, I worked with two different culture systems, an in vitro culture system based in hydrogels to keep a 3D culture system for the cells. And then I also work with ex vivo, which is directly with the tissue. Then I also work with different degenerative stages of cells. As I mentioned, healthy trauma cells and also degenerated cells. I also work with different osmolarities because it is known that in the disc, the osmolarity changes depending on the degenerative state. So I really wanted to make it as more physiological as possible, meaning that the trauma cells will be in a physiological osmolarity and the degenerative ones in a degenerative osmolarity. And basically, you can see here in the figure that I was treating them with IL-4, IL-10, IL-1 beta. IL-1 beta is a well-known catabolic factor, so I added to have kind of a positive control of catabolism in the disc, and then to compare also if it was similar to IL-1 response or not. And then I also combined it IL-1 with IL-4 and IL-1 with IL-10 to see what was these two IL-4, IL-10 doing in presence of IL-1 beta. So really quickly, I will also explain you what in vitro ex vivo means. In vitro, basically, we have two ingredients. We have the nucleus pulposus cells, and then we have the brown seaweed, from where we isolate the alginate, which is the polymer. So when we expand the cells, we have them in the, we need to expand them in monolayer because we need a huge amount of cells. And then we mix it with the alginate itself. Then we polymerize it in a, using calcium, and we end up having these nice beads that the cells are encapsulated inside, and then we put them with the culture in a culture system inside the hypoxia chamber because, as I mentioned, it's really hypoxic, the disc, so we wanted to keep it as physiological as possible. For the explant part, it's like we had this different system, which basically we collected tissue from the operations theaters. We took the empty tissue and we put small pieces, are called explants, inside silicone rings to avoid the excessive swelling of the tissue. And then we put it in the well plates, in 12 well plates in this case, and we also put it back in the hypoxia chamber and culture them. And after the culture, we applied the treatments. So now if we go a bit to the results, the first thing that we checked was the gene expression. And we, in vitro, we observed differences between the trauma and degenerative cells because the trauma cells were really responding to the IL-1 beta treatment. They were upregulating catabolic markers such as COX-2 or MMP3, the metalloproteinases. However, for the degenerative one, it was a mess. Like, it was a huge variability between the cells. The treatments were not really showing anything. And then we also thought that maybe the osmolarity could have an impact on that, not only the treatments, but after checking the degenerative osmolarity in trauma cells, we did not see any significant upregulation of catabolic markers, meaning that the real reason why they were upregulated in trauma cells was because of the treatment, and in the degenerative case, this kind of no response was not because of the osmolarity change. Then we also checked the gene expression in the explants, and again, it was a bit messy. We saw a really high variability in the response for the IL-4 treatment in the expression of IL-8, VEGF, MMP3, or aggrecan. We, on the other hand, saw that IL-10 seems to decrease it a bit, but there's still not a big difference. And of course, IL-1 beta was doing what we were expecting, increasing catabolic markers, IL-8, VEGF, or metalloproteinases, and decreasing the anabolic ones, the aggrecan. But the interesting part of my project, or the part that I think that is the best, I will say, is the secretome profiling that we did using proteomics that Christina really explained it. So perfect, I don't need to explain it, what it is. So basically, we collected all the supernatant from all these donors and all these culture systems, our different cells, and we ran Luminex to see what were the proteins that were secreted for these cells or these explants. And we mapped them and kind of create different profiles to see which ones were shared, in this case, the ones in the middle, and then which ones were unique to each culture system or cell degenerative state. Another thing that we wanted to do was compare the culture system, because of course they must have an effect on it. In the first case, we compared the baseline, so the no treatment group, in the ex vivo and in vitro settings to see if the system itself was changing something, and we saw some differences. For example, ex vivo, the macrophage migration inhibitor factor was a bit increased, well, significantly increased, in fact, and in the IL-2 receptor alpha was decreased. And then we did the same with IL-4 to see if IL-4 has different activities depending on the culture system, and again, it was increasing some factors and decreasing another ones. Then we have the whole screen of all these factors that I'm not going to go one by one, otherwise we will be here until tomorrow. But basically, we saw different things, especially with the trauma and degenerative in vitro experiments. The trauma ones really were reacting, as we saw in gene level, to IL-1 beta group. This kind of seems like opposite, since IL-1 beta is catabolic and here it's inducing anti-inflammatory, but it kind of makes sense since the cells, they are sensing this catabolism approaching, so they try to counter-react with these anti-inflammatory markers. In the case of the degenerative cells, all IL-1 beta only in presence with IL-4 or IL-10 was able to upregulate the secretion of these factors. We observe exactly the same in all the chemokines, trauma cells, only IL-1 beta was significantly increased in them. In the degenerative one, IL-4 and IL-10 enhance IL-1 beta chemokine secretion, because IL-1 beta group by itself, it was not able to do the same as in the trauma cells. They needed to have or IL-4 or IL-10 with them. And the same for the pro-inflammatory ones. Again, IL-1 beta really increasing them in the trauma, and in the pro-inflammatory ones, for the degenerative cells, it was the combination of both. And we could even see different responses, because in trauma, IL-4, do I have a pointer? No. Yeah. IL-4, it was decreasing a bit, for example, the secretion of IL-18, while in the degenerative, it was increasing it. So we really see that IL-4 has different roles depending on the cell type or in the cell stage. We also check angiogenic, neurogenic adhesion factors and regulators, and again the same. I'm not going to explain again and again the same. And the last important, or for me interesting part, was to check the extracellular matrix modulators, these ones that are in charge of recycling the extracellular matrix. And in the degenerative cells, we did not see any difference in the groups, like doesn't matter if it was not treated or if it was treated with IL-1, no differences, while in the trauma cells, IL-1 was really enhancing them, meaning that in the degenerative ones, they were already really activated since they were degenerative cells. So a bit as a summary, because all these graphs, I'm sure that you don't remember anything already, but basically, we can see that in trauma cells, IL-1 beta by itself can upregulate significantly at the secretion of pro-inflammatory, anti-inflammatory chemokines regulators or extracellular matrix remodeling. And IL-4 itself, it doesn't have any role. However, in the degenerated cells, we really observed that IL-1 beta, together with IL-4, was able to really enhance these pro-inflammatory, anti-inflammatory chemokine and adhesion factors, and even IL-10 was required for the significantly increasing the secretion of neurogenic and chemokines, meaning that IL-4 and IL-10, they have a catabolic role in the degenerative state. But of course, I cannot forget the explant experiments, because we also run proteomics for explants. And in order to do a quick overview, I just compare the IL-4 treatment with the no treatment, and only IL-4 by itself was able to significantly upregulate TNF, for example. Tumor necrosis factor is a catabolic marker, and there were other factors such as interferon gamma that was showing a trend to be significant too, which was really interesting. And then we also compare IL-4 and IL-1 axis, meaning that IL-1 by itself, it was not able to upregulate any secretion of these proteins, while IL-1, when it was in presence of IL-4, and we compare it with the IL-4 baseline, it was really increasing significantly the secretion of catabolic markers, such as IL-8 or T-weak. And again, a short summary. In this case, we already observed that IL-4 by itself, it was able to significantly upregulate pro-inflammatory markers. And again, with IL-1 beta, it was able to enhance the pro-inflammatory and chemokines, making IL-4 also in explants. I don't know why it's that slow. Also a catabolic. So in conclusion, we could say that IL-4 and IL-10 are catabolic mediators in human MP cells and tissue. We could also say that IL-4 possesses different roles depending on the degenerative stage of the cells, as well as depending on the culture system. And we'd also conclude that IL-4 could have a pleiotropic role effect in human IBD, since also has been reported to be pleiotropic in other kind of tissues or systems, such as the immune system. And with that, I would like to thank my supervisor and also my co-supervisors and the people from This For All. And if you have any questions, feel free to ask. Thank you.
Thank you very much for your nice presentation, Paola. Okay, already.
Nice talk. I have a question about the hydrogel you use. You use alginate, right? Mm-hmm. And do you do any functionalization on this hydrogel, or you use it as it is? Because my experience with alginate is the cell don't do much. They stay pretty round and they just survive. So do you think it's a good culture system for your cells?
Thank you for the question. So alginate system has been the gold standard in the IBD field, I don't know, maybe since 80s. So basically what I did, I cross-linked it with calcium in order to gellify it and keep the cells inside. And in general, for the nucleus pulposus cells, and I think also for the AF cells, it really brings a really nice environment for them, since it really mimics what it will be in the disc. They are like really entrapped by the extracellular matrix. They are only getting oxygen by diffusion, as in the disc, and the same for the nutrients. So they grow really nicely there, and yeah, they perform and they upregulate their own markers, let's say, because as I mentioned, first I expand them in monolayer, and in fact, I need to put them in alginate and wait 2 weeks to let them be in alginate, and then they recover the initial phenotype that they might lost in the monolayer phase, let's say. And also, it's an interesting question too, because there are recently also other kind of hydrogels, or let's say alginate modification that they have been studying. For example, I have been also studying a sulfated version of the alginate to keep it maybe more negatively charged and more native-like, let's say. And for me, it didn't work. All the cells died. So I was like, alginate, it works. If I start sulfating and modifying it, maybe not. But in cartilage, it works really well. So it will depend on the cell.
And also another quick question. How do you change the osmolarity? You just put sugar or PEG in the media, right?
Yes, so we had 2 approaches. One is increasing the osmolarity, adding sodium chloride. But of course, if you add that, you could also affect the sodium channels and all these things in the cells. And the other option that we had also was with an analog to kind of avoid the activation of this.
How do you explain that it has no effect on your...
It's really interesting because it has been published that they have an effect, but I did not see it. And I was really expecting it. And at least in trauma cells, the degenerative osmolarity was not really, or at least it was in my donors, it was not really increasing that much, maybe
in both systems, right? In vitro and ex vivo didn't have...
No, I only did it. No. So the comparison of the osmolarities, I did it in vitro, and the explants, I did them directly with the degenerative one because the explants came from patients with the intervertebral disc degeneration, so it was not really. But there are a lot of papers where they have been trying them, so I'm sure that if you check in PubMed, you will find much more information.
Thank you.
You're welcome.
Thank you so much, Paola. I have seen that for IL-4 signaling pathways, they usually involve co-receptors for the initiation of the signaling. And I don't know if you think that it could have an effect to have the culture adding, for example, IL-13, which is a co-receptor, and if this could change the answer of the cells.
Yes, in fact, that was the last experiment that I did in my PhD, because the next step was trying to track how this stimulation was in the downstream pathways. And we observed that sometimes they are like 3 different IL-4 receptors, and some of them, they require the co-stimulation with IL-13, as you mentioned. So I ran some experiments in alginate beads, co-stimulating IL-4, IL-13, and then by themselves. And I did not see at all anything, which was pretty sad, because I was thinking that it could kind of be the next step, but they were not really doing anything.
OK, thank you.
I also checked them in the degenerative cells, and I did not see anything either. So I don't know if it is also because the cell is already too much affect, I don't know. But it did not work at all.
And do you know if in the explants there's IL-13 present in the...
Yes, it was screened and it was secreted. It did not have any change depending on the treatments, but it was expressed. So they are able to express it, yeah.
OK, thank you.
Any more questions?
Yeah, Paola, thanks. So big highlight here is basically IL-4. So then the conclusion is there is no real conclusion. It might have a pleiotropic effect.
Yes.
Which means that we
Could
don't understand. It can be anything. So it's a bit philosophical question. So do you think that the expectation we might have about the specific effect of a molecule can be biased by knowledge that is not specific then to the system you're studying? Meaning, IL-4 is well known as an anti-inflammatory cytokine for the immune system. But actually, if you look down in the literature for intervertebral disc cells, it's very difficult to find evidences about that. So it means, so it can be a natural anti-inflammatory cytokine then for the immune system, but then is that true then for this kind of cells? And then up to which point do you think that knowledge can be biased?
Yeah, I mean, the thing is like when you try to create these models, you need to use the knowledge that is available. If there is not any paper reporting what IL-4 could do or not, you really need to go, for example, to the immune system and try to, or cartilage, to kind of take from there the knowledge. So that kind of could bias a bit this model. But on the other hand, I think that in the model, it was at least one article in the IBD that was showing the IL-4 was anabolic. So, and this person did like not the same studies like me, but it has the same cell type, the same treatment, and got a different result. So that's why I really think that IL-4 could have like these 2 different roles. And also, I really think that rather than be biased by other studies or things like this, could be also because we are not comparing the same things in terms of some experiments are done in monolayering or moxia. This totally can change the outcome. If it changed the outcome to change from trauma to degenerative or for in vitro to ex vivo, these kind of things really change it. And I think that if you don't take them into account and you just label it anabolic, no, it's anabolic under this environment. Maybe it's not if you change this environment, so, or other cell type, for example. So I really think that this could be really a big bias, and you really need to be able to identify it when you take knowledge from other papers or you want to contrast what you are doing.
So if you take that one paper on the intervertebral disc that shows that IL-4 has a or an anti-inflammatory effect or a pro-anabolic effect, so what do you trust more, your culture model or their culture model?
I mean, I will trust mine because I did it, and I know what I did. I don't know what they did. I am totally trusting them too, but it was not me in the lab. And I also think that the explanation is the environment bit of, because using this monolayer could differentiate the cells. Also, the normoxia could kind of affect all the pathways. So, and as we expected, IL-4 has an anabolic effect, so maybe it was what they were expecting. And it was what I was expecting, but with this data, I couldn't,
Yeah, because articular cartilage is a very different system in the sense that the articular cartilage layer is very close to the synovium that is highly affected by the innate immune system. So when we say, IL-4 then might help chondrocyte, etc., maybe that's also, there might be also an indirect effect.
Yes.
It actually helps the immune system that is screwing up a little bit less of the articular chondrocytes.
And also that there are different haplotypes of IL-4, so maybe because of the tissue in one, it's one more predominant than the other one, and that makes a really different outcome. That could also be another explanation.
Thanks.
Hi, thank you very much. Very nice presentation, and I'm happy that by coincidence I included some of the secretome analysis that we have performed. So this was totally a coincidence because I didn't have any clue of what you were analyzing here. Now, my question is because I'm now ignorant about IVD cells, and so what about mechanical compression? So in these models, so it's clear that to mimic really the environment. So have you done? Or anybody has, because in cartilage there was many.
Yes, so in my project, the mechanical aspect was not really contemplated. However, in Catherine's project, it was. And she worked not only with organ cultures and bioreactors, she also was able to prepare some cells inside hydrogels and in a small bioreactor be able to compress. And of course, this is something which is really common in the field, and a lot of people and it's totally shown that the mechanical loading, it really helps. Well, a healthy mechanical loading, of course, helps the phenotype, maintaining the phenotype of the cells, and also keep them more physiological. In my case, I did not have this included in my study, but it could be totally interesting to check what IL-4 could do. For example, in presence of mechanical loading, maybe it becomes again back anabolic.
Well, maybe you see any different.
Yeah, but I will need another PhD to go through this. So, thank you.
Thank you, Paola, for this very interesting talk. I have actually two questions. One was somehow a follow-up questions between the discussion and between Jerome and you. Because when I did at a higher level intervertebral disc research, I found that time is actually a really, really important factor. Meaning that IL-4 can be anabolic within the first, I don't know, I think you had 4 days or 2 days of testing, and afterwards it can change to catabolic. Or kind of what is your perception? Maybe you can't know because you didn't make long-term, over several kind of exposure times, but what would be...
I think it's the Zoom.
What? Yeah, well,
yeah,
but what would be your perception for time, or how would you interpret this?
So I think it's a really nice question because you can even see how important is the time in my project. As soon as you compare the gene expression and protein expression, due to the big amount of groups that I needed, I was not able to test different time points, so I was forced to measure everything in the same time. And I think that's why at gene level I did not see that much, because by then it was already, whatever response was already sent, and that's why I saw at the proteomic level something happening. So I would say that for protein expression would be a good time point, because it was 2 days, but it could be, for sure, it could be also the case that maybe it has different outcomes, the IL-4 or IL-10 stimulation, depending on the time. Maybe it could, we also saw, depending also in the degenerative state, it has different kind of roles. So it could be that it start having not a role, but if you keep it longer, and then IL-1 beta start accumulating, it changes. And maybe if I run the trauma cells, I keep them longer, and I put with IL-1, maybe suddenly they are reacting. That could, I would expect it totally, to be honest.
Okay, thank you.
We are done.
Okay, thank you very much. Please, let's close the session here.